Okay, Smack-on, good morning everyone. So today we are going to discuss about carbohydrates metabolism, okay? And then there are three processes here. So we go to the first one, which is glycogenesis, okay? The word here is glyco, refer to glycogen. Genesis is the synthesis. Okay, so glycogen synthesis. So glycogenesis defined as synthesis of glycogen from glucose, right? So usually glycogenesis, okay, occurs when blood glucose levels, right, are higher than the normal level. Okay, so we have excess glucose here in the blood. So this excess glucose, okay, will be converted into glycogen and stored in the liver and the muscle cells, right? So glycogenesis is regulated by insulin. All right, if you recall, insulin is secreted, okay, from the beta cells in the islets of the Langerhans in pancreas. And this glycogenesis process, right, takes place in the liver, right, hepatocytes. So now, basically, this is the flow of the glycogenesis, right? So now, without any further ado, let's start. Okay, so first things first, okay, hexokinase will catalyze, okay, an addition of a group of phosphate to this glucose to form glucose 6-phosphate, right? So addition of phosphate group added to the glucose catalyzed by hexokinase, and then we have glucose 6-phosphate. And then what happens to this glucose 6-phosphate is, right, phosphoglucomutase, right, catalyzes the rearrangement of the phosphate group, right, in the glucose 6-phosphate to form glucose 1-phosphate. So the phosphate group from the ATP, initially it was on the 6 carbon. Okay, so rearrangement to the first carbon. So we have glucose 1-phosphate. Next, all right, UDP glucose phosphorylase, right? UDP glucose phosphorylase catalyzes an addition of uracil, right, and a phosphate group from the uridine triphosphate to this glucose 1-phosphate to form glucose uracil diphosphate or UDP glucose. Right, so let me rephrase. UDP glucose phosphorylase, okay, catalyze the addition of uracil and a phosphate group from the uridine triphosphate into the glucose 1-phosphate to form UDP glucose. Right, now this UDP glucose, right, stands for glucose uracil diphosphate. Okay, and other name for this UDP glucose phosphorylase enzyme is UTP glucose 1-phosphate uridyl transferase, right, or glucose 1-phosphate how do you pronounce this, uridyl, is it, transferase. Okay, so I think UDP glucose phosphorylase is easier to remember. Okay, so now we have the UDP glucose. What happens to this UDP glucose? All right, glycogen synthase. All right, glycogen synthase will bind to this UDP glucose. Okay, and this will cause the UDP glucose to break down into oxonium ion and UDP. Or you can rephrase that as glycogen synthase catalyzes, okay, the breakdown of UDP glucose into oxonium ion and UDP, right? UDP, if you're wondering what is UDP, is uracil diphosphate. Now This oxonium ion, right, will bind with glycogenin to form glycogen. And then now we have the glycogen, but as you know, the glycogen, they exist as branches, right? So the formation of glycogen molecule is aided by glycogen branching enzyme, right? Glycogen branching enzyme. Now this glycogen branching enzyme transfers the end of a chain, right, or the end of the chain onto an earlier part. You can see from this diagram here, okay, via 1,6 glycosidic bond. So apparently this chain is being attached with this chain, okay, or this long chain here is being attached with this chain here, okay, via the alpha 1,6 glycosidic bond by the glycogen branching enzyme. So this will form branches, and these branches will further grow by addition of more alpha 1,4 glycosidic units. So that's about it, all right, on the glycogenesis. Now we further with glycogenolysis. Okay, glycogenolysis. Glyco refer to glycogen, all right, sorry, glycogen, glycogenolysis. Okay, glycogen refers to glycogen. Lysis means the breakdown of glycogen. So glycogenolysis is the opposite of glycogenesis. Okay, in glycogenesis you get glycogen. In glycogenolysis you get glucose. Alright, it's the breakdown of the glycogen. So now how does this process occurs? Right. So now first things first, alright, glycogenolysis is defined as the breakdown of glycogen into glucose. Okay, the reason is to raise glucose level in blood. Occurs in the liver when, okay, there are certain conditions that triggers glycogenolysis, right? So these two conditions here. So glycogenolysis are triggered, right, occurs in the liver when first the blood glucose levels are lower than the normal level, and this usually when the blood glucose levels, when they are lower in the blood, right, lower than the normal level, usually this condition is being accompanied, right, or it comes together with the decrease in insulin, and this occurs usually during fasting, right? So this condition will stimulate secretion of glucagon. Glucagon is a hormone. If you recall, it's being secreted. This glucagon is being secreted, right, by the alpha cells in the islets of Langerhans in the pancreas, right? Now another condition that triggers glycogenolysis is during fight or flight response, right? Okay, now during this fight or flight response, what happens? Epinephrine or adrenaline is secreted from the adrenal medulla, and the presence of this adrenaline will stimulate secretion of glucagon. Okay, now this glycogenolysis is regulated by glucagon that is secreted from the alpha cells in the islets of Langerhans in the pancreas. Now you've been wondering, how does glucagon plays a role here in glycogenolysis, right? So apparently, okay, the presence of glucagon, okay, it was being secreted by the alpha cells in the islets of Langerhans. So this glucagon will travel to the liver via the bloodstream. Once this glucagon enters, diffused into the liver, right, what happens? This glucagon will stimulate glycogen phosphorylase. Okay, and this glucagon also inhibit glycogen synthase, and this will begin glycogenolysis. if you recall, glycogen synthase catalyzes the breakdown of the UDP glucose into oxonium ions and UDP. So the action of this glycogen synthase is being inhibited by this glucagon, and this glucagon also stimulates glycogen phosphorylase and enzymes, right, that can catalyze, right, the glycogenolysis. Now, secretion of cortisol from the adrenal cortex only facilitates the activation of glycogen phosphorylase. Right, there are some books that mention this cortisol is needed, right, in the glycogenolysis. Yes, cortisol is needed, but in a way that this cortisol facilitates the activation of the glycogen phosphorylase. So this glycogen phosphorylase, right, will cleave this glycogen into glucose 1 phosphate, right, via phosphorylases. Next, phosphoglucomutase will catalyze the rearrangement of the phosphate group in the glucose 1 phosphate, right, to form glucose 6 phosphate. If you notice the position of the phosphate group in glucose 1 phosphate, right, was in the first carbon. So after the action of phosphoglucomutase, right, the position of the phosphate group changed to the 6th carbon. So now we have glucose 6 phosphate. So this glucose 6 phosphate, right, will be cleaved by glucose 6 phosphatase into glucose. So the glucose exits the liver to the bloodstream via GLUT2 transporter. This GLUT2 transporter can be found on the surface of the liver, right? Surface of the liver cells. Now, in the muscle, right, the lack of glucose 6 phosphatase, right, this particular enzyme, cause this glucose 6 phosphate, right, to be diverted to glycolysis, right, to be used for respiration within the muscle. This glucose 6 phosphatase, if you're wondering, it can be found abundantly in the liver, right? Then other than liver is the kidney and intestines. So now from here you can picture that a complete glycogenolysis, it occurs in the liver as well as kidney and intestine because the presence of this glucose 6 phosphatase, right? And the outcome of this glycogenolysis is glucose. And except in muscle, because muscle lack of glucose 6 phosphatase, so glycogenolysis, right, occurs until the production of glucose 6 phosphate, and this glucose 6 phosphate will be diverted into glycolysis, right, into glycolysis, and the product, which is glucose, will be used by the muscle itself. Right, now the third process is gluconeogenesis, right? So gluconeogenesis is defined as the synthesis of glucose from non-carbohydrate sources. Okay, now gluconeogenesis is stimulated or triggered when the blood glucose level is below the normal level during periods of fasting, starvation, low carbohydrates diet or high protein diet, right, and under condition of stress and intense exercise. So gluconeogenesis precursors are pyruvate from the lactate, okay, dihydroxyacetone phosphate from the glycerol, and pyruvate or oxaloacetate from amino acids, especially alanine. So now, what happens in gluconeogenesis? So anaerobic exercise causes accumulation of lactic acid. Okay, lactic acid in the muscle, and this lactic acid will diffuse into the bloodstream, okay, and reach the liver to be converted into pyruvate, or easier to say that this lactate will be oxidized into pyruvate, and this reaction is catalyzed by lactate dehydrogenase. Okay, now we have pyruvate here. Okay, next, amino acids, right? Elevated cortisol, right? Because as I mentioned earlier, this condition, right, because elevated cortisol, right, elevated cortisol in the blood. So this elevated cortisol is being secreted by adrenal cortex, and the presence of this cortisol will stimulate the breakdown of muscle into amino acids. Then transamination of these amino acids form pyruvate. Now we have pyruvate, right? So what happens with pyruvate? This process occurs in the liver cells, right? So this pyruvate, okay, will enter the mitochondria. Okay, now before I go into that part, right, so we already discussed about the presence of pyruvate, right, that came from lactate and from amino acid. Now the dihydroxyacetone phosphates, epinephrine and cortisol, right, will stimulate lipolysis of adipose tissues into glycerol. Then this glycerol undergoes gluconeogenic pathway to form dihydroxyacetone phosphates. Okay, so the outcome of this dihydroxyacetone phosphates, we go into that part later, because we need to discuss what happens here. Right, later the outcome of this incident here will merge with the dihydroxyacetone phosphate, which eventually marks the end of the gluconeogenesis, where we have formation or synthesis of glucose. All right, so the pyruvate will move into the mitochondria, okay, and undergoes carboxylation to form oxaloacetate. This is the Krebs cycle, if you recall. Okay, so let me rephrase. Pyruvate moves into the mitochondria and undergoes carboxylation, right, to form oxaloacetate. So this oxaloacetate, okay, will be reduced by, this oxaloacetate will be reduced into malate, okay, by NADH, and then this malate will be transported to the cytoplasm of the hepatocyte via malate shuttle. Okay, now once this malate is in the cytoplasm of the hepatocyte, this malate will be oxidized back to oxaloacetate by NAD+. Okay, so this malate shuttle, apparently for the sake of shuttling the oxaloacetate out from the mitochondria into the cytoplasm. Okay, now this oxaloacetate, okay, will be decarboxylated and then phosphorylated to form phosphoenol pyruvate. Okay, now this phosphoenol pyruvate, okay, undergoes reverse glycolysis to form glyceraldehyde 3-phosphate. Then glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate, okay, will be catalyzed by adolase to form fructose 1,6-bisphosphate. Okay, then fructose bisphosphatase catalyze hydrolysis of the fructose 1,6-bisphosphate to fructose 6-phosphate. And then phosphoglucose isomerase catalyze the conversion of fructose 6-phosphate to glucose 6-phosphate. Next, glucose 6-phosphate will be hydrolyzed by glucose 6-phosphatase, okay, in the endoplasmic reticulum to produce or to form glucose. Then this glucose will enter the bloodstream to increase blood glucose level to the normal level. Right? Now, this gluconeogenesis is important, okay, to maintain blood glucose level and avoiding hypoglycemia during fasting or exercise. Okay, and then this gluconeogenesis also removes lactate in the blood produced by the red blood cell and the muscle, especially during strenuous exercise. Okay, and then this gluconeogenesis also remove blood glycerol, which produced by adipose tissue. Now, the removal of lactate in the blood, okay, which is produced by muscle during strenuous exercise, okay, is being done by the Cori cycle. So in the Cori cycle, gluconeogenesis occurs. All right, it's a part of the Cori cycle. So this is gluconeogenesis. Right. Now let's proceed with Cori cycle. All right. Now Cori cycle is a metabolic pathway, right, which lactate produced by anaerobic glycolysis in the muscles moves to the liver, as you can see in this diagram here. Right, lactate, which is being produced by anaerobic glycolysis in the muscle during strenuous exercise. Right, this lactate moves to the liver via the bloodstream, and then in the liver, this lactate will be converted into glucose, which then this glucose will transported into the muscle by the bloodstream. And then in the muscle, this glucose again will be metabolized back to lactate, and the cycle continues. This Cori cycle. Now, what happens in Cori cycle? So, first things first, vigorous exercise, okay, will lead to an oxygen shortage, producing an aerobic condition in the muscle cells. Right, an aerobic condition in the muscle cells. Now, in order to regenerate NAD+, to keep the glycolysis going, if you recall, glycolysis needs NAD+, right? Now, in order to regenerate NAD+, to keep glycolysis going, pyruvate, okay, will be reduced to lactate in the muscle. So now, accumulation of lactate in the muscle causes muscle cramp. So, what happens? Right, to prevent muscle cramp, the lactate that is produced by the muscle will be carried by the blood, right, to the liver via the bloodstream. Okay, next, in the liver, what happens? This lactate undergoes gluconeogenesis. Okay, so first the lactate will be oxidized into pyruvate. Okay, so lactate to pyruvate is oxidation. Pyruvate to lactate is reducing. Okay. Now the lactate is oxidized to pyruvate, and then this pyruvate will be converted into glucose 6-phosphate. Then the glucose 6-phosphate will be converted into glucose. Right. So the glucose again carried by blood to the muscle. So the muscle use the glucose for cellular respiration. And then the glucose also can be converted into glycogen for storage in the liver. Remember, glucose is being formed here from the glucose 6-phosphate. So the glucose either go back to the muscle or store in the liver as glycogen. Okay, so importance of Cori cycle: prevention of lactic acidosis in the muscle under anaerobic conditions. So normally before this happens, the lactic acid is moved out of the muscles and into the liver. That's why when you do strenuous exercise, you don't get lactic acidosis, right? Because of this Cori cycle. So this Cori cycle also important in producing ATP during muscle activity. The Cori cycle functions more efficiently when muscle activity has ceased. So this allows the oxygen debt to be repaid, such that the Krebs cycle and electron transport chain can produce energy at peak efficiency. So that's why you can't continuously, okay, do your strenuous exercise. At one point you have to take a break, 1 minute to 3 minutes, right? The reason is for the core cycle to kick in efficiently. Okay, so that's it for today. Goodbye.